Okay, so this next section is on genetics. I think you will find it interesting. This chapter 10 is long. A lot of it has to do with Mendel, so we're going to learn about the basics of genetics first, and then we're going to expand from there. Mendel gives us what we know as basics. And there's still a whole section of genetics called Mendelian genetics, named after him and his laws that he founded, and the pea plants, and it's still considered like basic genetics and the basis of. So Mendel is considered the father of genetics. So the first thing we have to do is get through some of these bold terms. Heredity is just the tendency to be able to pass on traits. Okay, so if something is heritable, it means you can get it from your parents, or you can pass it on to your offspring. And so that's what we're talking about. We're not talking about things like being able to pass on your suntan or something like that. Maybe the ability to get one, but not the actual tan. And we kind of chuckle about that now, but that is something that they once believed. It's called the inheritance of acquired characteristics, because you can acquire these throughout your lifetime. And several hundred years ago, they thought that was possible, that whenever it was you would have a child, however you were at that point, you will pass that on to your son or daughter. So, you know, if you were working out, had really big muscles at that point, then you're going to give the big muscles to your son or daughter, because they thought that, and there's a name for it right now, I can't think of it, pangenesis, sorry. So it just came to me. So pangenesis, they thought that these little tiny things called pangenes, not the same as a gene. They thought that you would pass literally a tiny piece of every part of your body to your son or daughter. So literally a tiny chunk of your eye would travel from your eye down into your genitals or your womb, so that you could pass that to your offspring. Obviously, we know that that's not the case now, but they thought that. Another example of inherited traits, like passing it on, of acquired characteristics. There was a guy by the name of Lamarck, and he really championed that cause, that you could pass on these acquired characteristics. His example that he gave was the giraffe. And he said, well, how do you think the giraffe got such a long neck? The reason why is because they have to stretch for their food. And the longer they live, the longer their neck gets from stretching, and then they pass that on to their offspring. We know now that's not the case. But interesting nonetheless. And then there's all kinds of other things that they used to believe. So Mendel, in the 1800s, was seeking some sort of scientific basis for how traits are passed from parent to offspring. So he was calling the features characters. Okay, so for his pea plants that he was working with, flower color would be a character. Plant height would be another character. I don't tend to differentiate a lot between characters and traits. Your book does, where they're saying, you know, your hair color is the character. The trait you have is what color it is. So blonde or brown or red. For the pea plants, flower color is one character, but having either white or purple flowers, that's their trait. Whatever. All right. Keep in mind that Mendel, here's a picture of him. So he was a monk, remember? So he was a religious guy, lived in Austria, and spent an enormous number of years dealing with this stuff. This was also back in the 1800s, before we knew anything about DNA. They didn't discover DNA until mid-1900s. So it hasn't been that long ago that we finally figured out what it looks like and how it works. And we still don't know exactly how it works, not all the ins and outs. But when he was doing this then, he had no idea about DNA, no idea about chromosomes. He didn't know what went into that. We didn't know the steps of mitosis and meiosis. So he was really starting out doing this blindly. And, sometimes that leads to really good science when you don't have a background to work on. So when you start with a clean slate, you really are just taking a lot of notes and trying to intuitively put things together. And so that's where he was at with this. All right, so you heard me upstairs say he chose garden peas. And he did it on purpose. It wasn't just happenstance that he chose a good plant to work with. He looked for a plant that he knew he would be easy to work with and manipulate. So he said, first of all, peas are easy to grow. So he didn't have to worry about being a master gardener to be able to grow a bunch of peas. They're pretty small. He didn't have a huge plot of land to work with, so he was kind of short on space, so that's another good thing. The traits, says varieties have many distinguishable traits, like I said, that come in only 2 varieties. Like the flower color that he was working with, it's either white or purple. There were no other colors. The peas are either yellow or green. The plants are either tall or short. So he was purposely working with these plants because of that. The reproductive organs. Okay? For plants that flower, their reproductive organs are in the flowers. So because of that, they are very easy to control in terms of which plants he wanted to cross, which he wanted to self-fertilize. That's something different that we can't do, that plants can. In each flower, you have that center, it's called a pistil, that pokes up, and then you have all of these shorter stems that have the pollen on them. Those are all the stamen. The pistil is what holds the egg. Now the egg actually sits down here at the base of the flower. And so any sperm that fertilizes the egg has to land at the top of the pistil and then migrate all the way down to the bottom there. The pollen on the stamen, that contains the sperm. So because any one flower has its own sperm surrounding where the egg shaft is, it can self-fertilize. Ideally, they don't always self-fertilize because for evolution, it's good to have a lot of mixture of genes. And so that's what we need, for example, bees for. Bees land on one flower and go to another, many times of a similar, same species plant, but a separate one. So that way they can take pollen from one and put it on the pistil of another, and that's how you get cross-pollination. There's also an entire field of study that deals with how plants, the evolutionary mechanisms that they have evolved to cross-fertilize and spread, things like that. So a good example of that are called samaras, the helicopters from maple trees. It's called a samara. And that evolved so that when they fall out away from the tree, and that way new seedlings can sprout up away from the parent plant. Cockleburs, so you have to pick them off your pants and stuff after you walk through the woods. That's another evolutionary mechanism that has evolved where they will stick to any passersby, animals, humans, whatever, and then hopefully get dropped somewhere else. Fruits and berries. Okay. So the seeds within can go through an entire digestive tract and then be pooped out somewhere else. So the birds that are eating all the berries, they eat seeds, and then they fly off somewhere, and the seeds get dropped with their poop or their droppings. And I guess an added bonus is the poop probably helps to fertilize it. So there's lots of good things about these plants. The peas themselves, because they're short, he didn't have to climb trees to try and get to the flowers. He was able to go from one plant to the next and easily decide whether he wanted to self-fertilize that plant or if he wanted to cross this plant with this plant. And then he would actually do it with scissors and a paintbrush. Let me get to so that's what they're trying to show you here in this picture. I wish it was bigger. I thought they had a bigger picture. I'll just use my book and put it up there. So let me show you what he was doing, because it's pretty crazy that he spent this much time. He had to have a lot of patience. And so first of all, they're trying to show you what the flowers look like. And so what they're showing you here, there's different names than I've used, but this is also called the carpel. Notice that's the female part. The egg is going to sit down here at the base. The anthers, these are the stamen that I talked about. The pollen is the yellow stuff that sits on top. If that pollen penetrates that carpel and makes the journey down, then it self-fertilizes. Otherwise, the pollen from this flower can be blown, say, even in the wind and land on another flower and fertilize that. What he did is he wanted to choose which would mate with which. So he took literally a pair of small scissors and a fine paintbrush so that he could go from one flower to the next, and he would cut off the pollen before it forms, actually. So he would cut off the stamen or the anthers. And then on another plant, he would wait for, if that was his source of pollen, he'd wait for the pollen to develop and then take the paintbrush, dip it in the pollen from one flower, and go to the other one and fertilize it. Insanely time consuming. I just can't imagine. So he was constantly watching, because before the pollen forms, he would want to cut it off, because the action of cutting this off would kind of make the pollen go everywhere. So this was done before then. If there were any that he wanted to self-fertilize, then he wouldn't have to do that. He would just take some little bags, and he did this too. He would bag the flowers once he was done to make sure that no outside pollen was able to get in. So if it was self-fertilized, he could just bag the flower and maybe shake it so that the pollen gets on itself. So that gives you an idea of what this guy was doing. So to start off, he wanted to make sure, so once he had the plants picked out, he wanted to make sure that he had what are called true breeding plants. True breeding means when they self-fertilize, the offspring will be the same every single time, so that there are no surprises. A true breeding plant will always, always, always give the same offspring. Think about their genes then. What two alleles, what would their genes be? What would their genotype be? From upstairs in lab. If every time they mate, every time they self-fertilize, they always give the same thing, then their two alleles must be identical. So that means that we're talking about flower color. That's always easy because we can identify with whether the flowers are white or purple. What he was looking at, okay, I have this purple flowered plant and I have this white flowered plant. He's going to plant them. He's going to get them to grow up to be adult plants, and then he's going to allow them, he's going to bag the flowers so that they always self-fertilize. Once they self-fertilize, then he takes good care of them. You have to wait until they produce seed. Okay, so those are the peas. And then he's going to harvest the peas, and he's going to plant those, and then wait for those plants to grow before he can finally see what the offspring show. Okay, so, like I said, he did this for a long time. That would take months to make sure that his first beginning plants are true breeding. But here's what he did then. If purple always gives purple, and white always gives white, then their genes, the two alleles, must be the same. So we're going to say big A is purple and little a is white. So the purple flowered plants must have been this if they're true breeding, because remember, through meiosis, they don't give both alleles, they only give one. So if it's the same thing every time, then it makes sense that both alleles must be the same. Same thing here with the white ones. So that's what he meant. That's what it means here, that it's true breeding. Once he did that and he was satisfied that those plants would always give the same thing, that's when he would start doing his crosses and investigations. The starter plants are called the P generation. Those are the starting plants. Think P for parents. The P generation, those are the parent plants. They are always true breeding for Mendel. Always true breeding for Mendel. There's a term for this. You heard me drop it today in lab. When both alleles are the same, we call that homozygous. It's coming. It's on another slide. Homozygous means both alleles, both the letters for that trait in that one plant are the same. That's homozygous. We haven't seen one yet, but there's another one that's called heterozygous. And that's having one of each allele, like big A, little a. That would be heterozygous. That's less predictable. If he has a plant that's heterozygous, that means that it's not always going to give the same allele. So that's not as predictable. That's not what he wanted. If he had a plant like that, he might keep it for other study purposes, but he's not going to use it in P generation. Okay, now, he looked at least 7 different traits. Flower color was one of them. So every time he looked at one, he had to do this again. So flower color, flower position for pea plants. The flowers are either at the end of a stem or they're called axial, which is where 2 branches come together. He did that for height, tall and short. He did that for pea color, green and yellow, pea shape, smooth, wrinkled and round. So 7 things he had to do this for and make sure that the pea generation was always predictable. Okay, so true breeding means homozygous, same thing. Okay. Then, once he was ready, he would take 2 parent plants that are different in one way. So these 2 plants that are different for flower color, everything else about those plants is the same. So that means one has purple flowers, the other has white flowers. Other than that, they're both tall, they both have green peas, the peas are wrinkled, the pods are green, they are constricted. Everything else is identical. The only thing that's different is the flower color. And he would cross them. That's when he would allow them to mature, but watch the flowers so that the pollen doesn't form yet, and then he's going to cut off the pollen and transfer one to the other and put bags on. Okay. Then they have to grow, they have to produce the peas, then he's going to harvest the peas and plant them. Those plants are the F1s. F1N actually stands for first filial. That's the kids. F1 is first filial. That's the kids or the offspring of the P generation. So the P plants, those are the parents. Their kids are the F1s. And then he would take some of the F1s and allow them to self-fertilize, which is the same thing as crossing 2 of the same plant. And their kids are the F2s. And so you start with the P generation, their offspring are the F1s, and the F1's offspring are the F2s, and we could keep going. F3, F4, even on a human pedigree chart, you can name it that. where as far back as you go, you would start with the P generation and then F1, F2, F3, F4, F5. And he did go further, but it's these three, the Ps, the F1s, and the F2s, it's those three generations that really gave him most of his useful information. The F3s actually give us more information now. Okay, so we still have his notes, but in the F3 generation, it started to get really muddy because of what we now know about genetics that he couldn't explain back then, and that is, meiosis and the fact that in meiosis the chromosomes cross over and swap pieces, etc. Since he didn't know about chromosomes, now he could see them. The microscopes were good enough back then that he would be able to see them on a slide, but other than that, they didn't know about PMAT. All right, so we already looked at that. A few more terms. Okay, so, and also kind of what's going on at the same time. Here's what happened with Mendel's stuff. Once he started with the P generation, okay, let's go back to flower color. He takes a plant with purple flowers and he crosses it with one with white flowers. In the F1s, in their offspring, they all were identical. For this cross, they all ended up with purple flowers. Okay, so we're taking a purple plant times a white plant. Okay, so this is the P generation. In the F1s, nothing but purple. That tells him which one is dominant, and it's purple. So the dominant trait is the one that is called expressed. That's the one that's expressed in the heterozygote. What's that? Well, the heterozygote is the organism that has one dominant allele and one small allele. Because if you think about the genes of what's happening here, the purple flowered plant is big A, big A. Because it's true breeding, we know that both alleles must be the same. The white flowered plant is little a, little a. I'm choosing A for both of those because it's the same trait we're talking about. Does that make sense? If you're talking about flower color, we're going to make A our letter. There's only two colors, which is good. So we have one that's dominant. I'm going to make that a capital A. That's the purple. And one that is recessive, not dominant. That's the white one. I'm going to give it the lowercase a. If there were a third color, we could still use A, but I might put, say, a line across the top or something to show that it's a third allele. So when you guys are doing your Punnett squares in genetics, for any one trait, pick one letter and choose wisely so that the uppercase and lowercase don't resemble one another. Now, after meiosis, what does the purple plant give? Always a big A. So the sperm or egg from the purple plant will have one A, but it's a capital letter A. It's a dominant one because that's all it has. For the white plant, the same is true, except it always will give a small a. So in these F1s, what's their genotype? They are all big A, little a. That's why they look purple. Now, Mendel didn't know about this big A, little a stuff yet. Keep in mind, he was only observing color and then had to try to figure out what was going on. Because of all of the crazy hypotheses out there, there was, the theory of acquired characteristics, and there was pangenesis, and there was another one that I can't remember the name, but it was blending, where at first it kind of makes sense, especially when you look at humans and in families. You think, oh, well, it's kind of a blend. The offspring are kind of a blend of their two parents. But if that's true, If blending is true, eventually what happens? Wouldn't everybody start looking the same? If you keep blending and blending and blending. Same thing for any plants. If blending were true, we wouldn't get purple flowers, right? What would we get? What would we get? What would you get if you crossed purple and white and blended it? White, purple, right? And so this shows, what he did right here is show that it's not blending. He took a purple flower plant and a white flower plant that he tested first to make sure that they would breed true. And if blending were the case, then their offspring should give light purple flowers. They're not. The purple in the F1s is just as dark as the purple in the pea plants, the pea generation pea plants.
Wouldn't a recessive tree pass down long enough to become a dominant tree?
That can happen, but it doesn't have to do necessarily with time. It has to do with mutation. And we'll talk about that later. All right, so keep that in mind. So he crossed those, got all purple. His next step was to self-fertilize the F1s. Because what he wanted to know is, okay, they're all purple. Is there anything still there that would code for white? Is the white completely gone or not? And the only way to test that is to take these F1s and self-fertilize them maybe several times to see if he gets nothing but purple after that. In which case, the white would have been eradicated the first time around. So that's what he was testing. So when he self-fertilizes the F1s, their offspring are the F2s. And what he saw in the F2s was that he got purple and white. So the white came back. If you were looking at that, what would that tell you? Got a purple and a white, and I crossed those, plant their seeds, I got nothing but purple. I self-fertilize those, plant their seeds, and voila, I now have purple and white again. What does that tell you about these F1s? They look purple, but what? They still carry something for white. So that's what it told Mendel too. He doesn't know what, he doesn't know how. They look purple, but they must carry something in them for white, because otherwise their offspring couldn't possibly have white flowers. Now here's their proportions. 100% purple for those F1s. In the F2s, every time he did this, the numbers came out about the same. And that is, what do you think? Three-fourths to one-fourth. Or 3 to 1. The ratio every time came out to be 3 to 1. And it doesn't matter if he was talking about flower color, because after he was done with flower color, he would move on and he would look at maybe plant height. And he would take a tall one that always breeds true for tall, and a short one that always breeds true for short, cross them. All the F1s would be tall, because that's the dominant trait, dominant allele, I should say. And then self-fertilize those, and he would get tall and short. But the proportion, the numbers are a big deal, because it would always be a 3 to 1 ratio. So that told him that not only do they still contain that recessive something, the recessive allele is what it was, but mathematically there's something going on here too. Okay. So dominant, one way to give a definition for these, the dominant trait, recessive trait, or dominant allele, recessive allele, It's when it's heterozygous. When you have an individual that's heterozygous, the dominant trait is the one that shows or is expressed. The recessive one is hidden. We don't see it. Here, it took a long time, but eventually they found his notes. I can't remember where, but stashed away, and it was long after his death. So that means that when Mendel was finished and wrote everything, there was no notoriety for this. He got nothing. So many years after his death, I would say, I don't know, 20, was when they started, first of all, they had to find it. And then even when they first found it, they kind of thought it was rubbish. Thank goodness they didn't throw it away. It wasn't until they really started getting into genetics at the beginning of the 1900s that they realized what he had done. So this shows his actual numbers. And I'm always awed by this too, because he actually had to count these. So the flower thing, purple flowers versus white flowers, he counted in the F2s 705 plants that had purple flowers and 224 plants that had white flowers. And so that's about 1,000 plants. Check out the next one. The peas inside. When he crossed just for the color of the peas in the F2s, he had to open every single pod and count the peas individually. He counted 6,022 yellow ones and 2,001 green ones. And so that's what this man was doing. That's pretty motivated to continue to do this day in, day out by yourself. I don't know, I haven't ever read his story to know. You would think he would have maybe somebody working with him, but it kind of depends too on what type of monk he was. Because if they had a vow of silence or something, I don't know, then that would play into it. Anyways. The important thing is in that ratio, because over here, they do the math for you and they show you that every single time it's approximately 3 to 1. All right. Then he started trying to figure out why and what was going on. And he used different terms because there wasn't yet a term for genes. There wasn't a term for alleles, no chromosomes, no DNA. So he was trying to explain what was going on. Trying to remember, it's in your reading. Anybody read yet? It'll come to me. What he called, he had a different term for genes. It'll come to me. Anyways, this goes on to say that he did self-fertilize the F2s to form the F3, but he found that some of the F2s were true breeding, some were not. Some carried the white allele, some didn't. So he couldn't explain what was going on. But what he did find was that every time he ever dealt with a white-flowered plant, or for any of the other 7 traits, the recessive form, he found that the recessive form was always true breeding. So for flower color, white is recessive. And he could always count on the white ones being true breeding, so that when they self-fertilized, they never, ever, ever did anything but white flowers. It's the purple ones that he couldn't count on. And so when he self-fertilizes the purple plants, some of them are true breeding and always give purple, and then others aren't. And that's what was throwing him off, was how could he possibly tell from a purple flowered plant which one is going to be true breeding and which one is not, and why? That bothered him too. Well, how come some are and some aren't? Well, if you don't know what genes are, factors. We call genes factors. Because there was no word for gene, so there must be some factor, he said. The white factor, the purple factor, whatever. So there was a ratio with the purple ones too, though, and he found that 2/3 of these were not true breeding. 1/3 of these were true breeding, pretty much every time. We can explain that now because of the genetics. We know now what happens here. If we cross 2 of these, then you're going to get some purples that are big A, big A. Well, those are the ones that are true breeding. You're going to get others that are big A, little a. Those are the ones that are not true breeding. And then, of course, the white ones are always little a, little a. So now it's fairly easy for us to understand and to explain. But for him, of course, not having any knowledge to work off of in the first place, it wasn't. Okay, the ratio stuff. What he was doing with this whole setup for any one of those traits was called a monohybrid cross. And that's when he crossed or self-fertilizes the F1s. Looks like this. A monohybrid cross. Mono means what? One. What does hybrid mean? Yeah, some sort of combination of 2 different things. And we use it loosely in a lot of ways, like hybrid cars feed off of gasoline and electricity. Okay, so they're a hybrid. Well, the plants that are hybrids are the F1s because they come from crossing a purple with a white. So the F1s, remember, whoops, not FI. The F1s for flower color were purple, genetics-wise. Their genotype is big A, little a. Phenotype is what it looks like, or the outcome of the genes. Genotype are the letters. Those are the genes for the alleles. So when we set up a Punnett square, remember that these are big A, little a. What we're really doing, think about, we're not crossing the plants, we're really just crossing their sperm or egg. And their sperm or egg doesn't have 2 A's in it, only one of these. So what we're saying with this heterozygote, it's either the sperm or egg is going to have a big A or a little a. Not both because of meiosis. The other one is also going to have a big A or a little a. Then when we fill in those boxes, just like we did in lab, this is where we're going to see Mendel's ratios. He wasn't doing Punnett squares because, remember, he didn't know anything about genetics. Its ratios, remember, in the F2s, 3 purple to 1 white, that's right here, 3 to 1. So the phenotype ratio is 3 to 1. The genotype ratio (1 to 2 to 1). You have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Homozygous means the 2 alleles are the same. It can either be homozygous dominant, big A, or homozygous recessive, little a, little a. Heterozygous is not dominant or recessive. There's only one type, big A, little a. So these ratios remember, come from phenotype for Mendel. So a phenotypic ratio is looking at the outcome of the genes, the expression of the genes, or what we can see. So 3 purples to 1 white, that's what this is. 3 purples to 1 white. Or 3/4 to 1/4, right? 75% purple, 25% white. The genotypic ratio, that's when you do look at the letters. We have 1 that's homozygous dominant, 2 that are heterozygous, and 1 that's homozygous recessive. So 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. These you have to know. 3 to 1, 1 to 2 to 1. You have to know those numbers. It should automatically make you think of this Punnett square, where you're crossing 2 heterozygotes. They're heterozygous just for 1 condition, so 1 letter. If I tell you 2 individuals were crossed for this 1 trait and 75% of them are black and the other 25% are white, you should know automatically, oh, well, the parents are big A, little a. Or for a genotypic ratio, I could spit something out and say, the genotypic ratio of this is 1 to 2 to 1. Right away, that should tell you, oh, both parents are heterozygous. Okay? This is called a monohybrid cross because he's crossing 2 hybrids that are hybrids in for one trait. A hybrid is a heterozygote. But here we're only looking at 1 trait. We're just looking at flower color. We will get to where we're looking at 2 traits at a time, but right now it's just 1. That's why it's a monohybrid cross. And these numbers are always associated with a monohybrid cross. So if I ask you on the test, what's the phenotypic ratio for a monohybrid cross, you're going to tell me 3 to 1. What's the genotypic ratio of a monohybrid cross? 1 to 2 to 1. Okay, if you like pictures, here's what he was looking at. Here's the 2 parents. Cross those, you're always going to get plants that have nothing but purple flowers. Self-fertilize those, and this is what he got. Mostly purple, 75% of the plants have purple flowers, but 25% have white. So he knows that these F1s still carry that white factor, is what he would call it. The white ones, always true breeding. The purple ones, some are and some aren't. This one's true breeding. These are not. When self-fertilized, they'll give that same 3 to 1 ratio. That's because these 2 are heterozygous, big A, little a. Questions so far? When you cross-fertilize, you're crossing 2 different plants. Separate or individuals, maybe not plants, it could be animals too, but 2 individuals that are different for whatever trait you're looking at. That's a cross. I guess they don't have to be different. You could still say crossing 2 individuals. Self-fertilize is the exact same thing as crossing 2 that are identical. All right. Mendel's factors we know are called genes. We've already gone over homozygous and heterozygous. You remember that homozygous means both letters are the same, either capital or lowercase. In reality, what that means, both of their alleles are the same. And if you think about that further, where do we find alleles? What are they? They're genes. Where do we find genes? DNA. And where's the DNA? in the nucleus. In the somatic cells, what's a somatic cell? Non-sex cell. So in all of our body, except for sperm and egg, we have 46 chromosomes, 23 from mom and 23 from dad, which means when we're talking about alleles, they occur in pairs because of the chromosome pairs. So if we were talking about some sort of human trait here, the big A comes from mom and this one comes from dad. They actually are on separate chromosomes. It could be maybe chromosome number 3. We have one copy of number 3 from mom and one copy of number 3 from dad, and it just so happens that they both carry a big A. Okay, so you have to try and link that together in your brain, because when it goes through meiosis, that's when this gets separated, because the chromosome pairs get separated from one another. When they're the same, not a big difference, because the outcome will always be the same. It's when you are heterozygous that you can get different things. Furthermore, when we start looking at multiple traits at the same time, then it gets messier yet. All right. You guys can look through his hypotheses. I'm not going to ask you about that. I will ask you about his laws, which we're getting to. There's alleles, okay, alternative forms. Now, he did get that far. He knew that there were these factors, genes, and he further deduced that there must be alternative forms. Well, obviously, otherwise you couldn't have different colors of flowers. He just called them alternative forms of a factor. We call them alleles. And we already know this, the genotype, those are the alleles. We represent those as letters. If you think about what they really are, an allele isn't a letter, right? It's a section of a chromosome, so it's a section of DNA that reads some nucleotide sequence. A, T, C, G, G, T, whatever it might be. That's really what we're talking about. But that's going to define for that cell what protein to make. And it's the proteins that make the traits. For the purple flowers, they have some allele, whatever this capital A is, that makes proteins that are a pigment. And we just finished photosynthesis. You guys know that a pigment is any molecule that has the ability to capture light and reflect some. Okay? The purple pigment is a protein that reflects in the purple wavelengths. That's why it looks purple. For the white flowers, they don't make the pigment. That's why they appear white. So really, this little a is the absence of any pigment. That they don't make a protein for that. If there would be, say, in pea plants, a third color, I know that there are some that are pink, that may be yet a third allele. That's a different pigment. So when you think about traits and when you think about alleles, you will best understand it if you link all that together. That the letter represents one section of one chromosome. If that section on the homologous chromosome is the same, then that's when we get homozygous, here or here. When that section on the homologous chromosome is different, that's when it's heterozygous, big A, little a. All right. Some of these hypotheses, well, I'm not going to test you on it. It is kind of interesting to read back like what he came up with. For example, the two alleles do not affect each other. They didn't in the pea plants. When we really got into genetics later in the 1900s, we found that sometimes they actually do. For example, I'm trying to think. Do you know what Indian corn looks like? Popular in the fall, usually dried, and it has all the different colors of kernels on one corn cob. So, you know, something that is really, really dark blue or almost black, the yellow, you might have a brown or a reddish. The colors of those kernels are not due to one gene, but two. And so when you have the dominant form for both of those genes, that's when you get the most pigment. So you get the black kernels. When you have the dominant form of only one, then you might get the brown or the red kernel. When you have the dominant form for the other, that's when you get the yellow. When you have the recessive for both, you get the white kernels. And so in that case, they do interact with one another, but he didn't see that with pea plants. Here's some human stuff, just to keep it interesting. Recessive traits, now this is just simple Mendelian stuff, meaning that in order to have a recessive trait, you have to be homozygous recessive. Common baldness. In common baldness, people start losing their hair from the front rather than on the back top. And it's not just men, women can suffer from this too. If you look at your grandparents or great-grandparents, right, most everybody who gets old starts to get thinning hair. And a lot of times it is from the front, a lot of times it is on the top. And so common baldness, that's what they're talking about, M-shaped, is here in the front. Albinism, what's albinism? Yeah, albino, lack of pigment. Complete lack of melanin. I mean, if they're truly albino, these people don't have any melanin. So they will not tan no matter how slowly they would, try to introduce themselves to the sun. They won't tan because they don't have the ability to produce the melanin. Color of their eyes. You're on the right track, though. Even if you have, say, an albino guinea pig or something, what color are the eyes? Red, right? Or pink? You know why? What are you seeing? You're not seeing the colored iris like we have. You're seeing, because there's no, the eye color that you get, that's from melanin production. So people who have less melanin, that's why they usually have lighter colored eyes, and that's why they typically burn easier. It's all tied together. In those who are albino, they don't have any melanin, so their irises don't have any color, and what you're seeing are the blood vessels in the back of their eye. That's why it looks pink or red. Cool, right? Alkaptonuria. This doesn't mean a whole lot to you. Honestly, I couldn't tell you what the phenotype is for this, other than it tells you the inability to metabolize homogenistic acid. That's an amino acid. We can't, our bodies don't make all 20 amino acids. Some of them we do need in our diet, okay, like possibly homogenistic acid, but people who have this, that would kill them if they keep eating that. There's other conditions like that. Oh, they don't have it on here. There's another one that's called PKU. It's called phenylketonuria. If anybody has, you have a bottle. I know on some diet sodas, it will say on the side, and it's always in small print, attention phenylketonurics. This product contains phenylalanine. Like, I know Diet Dew has it, maybe some others, but they have a similar condition, but instead of homogenistic acid, they cannot metabolize phenylalanine, which is a different kind of amino acid. And for them, the outcome would be permanent brain damage. So that tends to be something, every state, when you guys start to have kids, keep that in mind, but every state has different tests that they automatically do on newborns. If you want more or fewer, you have to be on top of the game enough to have already done your research and know what you want to request ahead of time. Otherwise, you're just going to get whatever the normal is. And so with some states, it's as few as maybe 15. Other states, they might do 50 because it's a state process. So you have to know ahead of time what they're going to test for. But one of the things that most of them test for in Illinois does is PKU, because it's a fairly standardized test, and kids with PKU, when you're born with it, it doesn't necessarily mean you're doomed to have brain damage. Phenylalanine is something that you can avoid in your diet. And so that's what, if you have a kid that has PKU, you just make sure that they don't ever get phenylalanine. And so it's easily avoidable. Can't fix it. They still have the DNA. They still have the genotype. Okay? It's recessive, so they would be little a for that. Which means, what does that tell you about the parents? Or both carriers, heterozygous. Because these little a's have to come from somewhere, right? One comes from mom, one comes from dad. So normally, people already know that PKU runs in the family. Not always, but normally. And so they would know that if their state doesn't test for that, they're going to want that done. Anyways, red-green color blindness. This is not the only kind of color blindness. There are actually many different kinds of color blindness. This one is recessive, and it's actually carried on the X chromosome, which they're not mentioning here. But the red and the green to these people look the same. They can't tell. So like with stoplights, they can't see red or green. They just know what the order is. And if the top light is lit up, they know that's stop. And if the bottom one is lit up, they know that's go. And to them, it looks like various shades of gray or tans. Dominant stuff. If you have a dominant trait, most likely you are heterogeneous, heterozygous. Some of these dominant things, it's not on there. There are some dominant diseases, which we'll get into later, that will kill you in the homozygous dominant form. Now, you would never, it's not like somebody would know that they're going to die from it. But what happens to a zygote in humans, one of them is, say, Huntington's disease. That's when mentally you start to deteriorate. Typically, it's after, say, age 35. So it is something now that we can test for, and you would know that it runs in your family. But nobody living that we know of has ever been homozygous dominant, because that's lethal. So that in the zygote, if the sperm and the egg each carry a dominant allele, when that fuses at some point, the female's body knows that there's something wrong and it miscarries. All right, other things, mid-digital hair. They're talking about right here. Sorry, I did not mean to use my middle finger at you. Okay, so right here, I don't have any. It's never really, and this is stuff that maybe you've never even thought of. Short fingers. Now, truly, for brachydactyly, you'd have to look it up, because everybody's going to have different lengths anyways, and that's related typically to your height. But there is a condition where it would be unusual, like your fingers would be unusually short for your height. PTC sensitivity, meaning you can taste it. And we'll do that within the next few weeks in lab. And some of these are weird that it would be genetic. Like, why would that be a genetic thing? Some of these are easier to understand than others. With PTC, because maybe I'd have to look up where this stuff actually comes from if it's present in nature, but they think that the ability to taste some of these things might have been a mechanism to avoid poisonous substances 1,000 years ago, or whenever it was. They think that's true also of morning sickness in pregnant women. They think that if they are not feeling well all the time, that may decrease the chances that they would eat something that has an extreme taste. And a lot of toxic substances fall into that category of extreme taste. They think maybe that's why it continues. Because if you think about it, way before, way back with cavemen and tribal living and stuff, why would morning sickness continue to be passed on if it's a genetic thing? Because you'd think that maybe those women, a lot of them, wouldn't eat enough or sustain themselves enough. Maybe they would get sick and die. Maybe their infants would get sick and die. Maybe they'd miscarry because they weren't eating, whatever it might be. And so the question is, if that's the case, then they would have fewer kids, and that trait really should get passed on less. But we still see it all the time. Well, they think it has a lot to do with those moms who didn't ever develop morning sickness probably had a higher incidence of eating something that would be toxic to them or their child. Camptodactyly, inability to straighten your little finger. I mean, some people just cannot straighten their little finger all the way. Polydactyly, extra fingers and toes. These aren't usually full functioning fingers and toes. And it's really more common than you think. Some of you might know somebody who actually has this, and usually it's kind of a nub. And so, and most often it'll be fixed. It's more commonly, fixed surgically with the hands. Just not because it poses any health risk. That's just a societal thing, so that the kid doesn't get made fun of for having some little extra growth on their hand. On their feet, they less commonly worry about it because people don't see your feet as often. But again, it's usually like a smaller, a teeny little nub of a toe. All right, back to Mendel stuff. We already talked about that. Punnett squares. This is just bold, so you know what it is. But for a definition, that would be like a chart showing the possible outcomes of a cross. A Punnett square is a chart that shows possible outcomes of a cross. A chart that shows possible outcomes of a cross. When you do a Punnett square, keep in mind that you are crossing two individuals' sperm or egg, their gametes, not the whole individual themselves. That's why if we were doing a cross with this individual that's little a, we wouldn't have to include both of them because he or she, or it, would give a small a every time. So in their gametes, they would always carry a small a. That's what we're crossing in those Punnett squares, are the gametes of the individuals. And here's another look at what we just did. P is not a good letter to choose. You can see how confusing that might be unless you're really careful about whether you're showing capital or lowercase. But this is just like our big A, little a cross over there, where for each one it's 25%. I usually lump these together. 50% are big P, little p. Here's some more, but with the colored flowers inside. So here's how we see the parental generation. True breeding for purple means big P, true breeding for white, little p. Everything in here is 100% heterozygous, big P, little p, all purple. Crossing two of those, that's what they're showing here, big P, little p here, big P, little p here. This is the important one because that's where we get the 3-to-1 phenotype ratio and the 1 to 2 to 1 genotype ratio. So that's a monohybrid cross. All right, this one's kind of interesting. Mendel was the first one to come up with this, and we still use it today. He devised what's called a test cross. He called it a back cross. They've now changed it to a test cross. And here's what he did. He was interested, okay, how can I possibly know for the F2s? Remember the F1s were all purple? And the F2s, some of them were purple and some of them were white. The ones that are white were always true breeding. The ones that were purple weren't always. So it's these purple ones. He can't tell by looking, are they true breeding or not? So he wanted to know, is there a way that he could do some crossing to be able to tell right away for any one purple plant without having to breed it several times before he can be sure it's true breeding. one cross would be a lot easier and a lot faster. So what he decided was, well, since the white is always true breeding, I can take a white flowered plant and cross it with a purple one to see if I get any white flowered offspring. And that will tell me right away if that purple plant carries the white allele or not, the white factor. So he called it a back cross because he would take this F2 purple and he would back cross it to the P generation white one. So he would take this purple one, he's going to back cross it to that white one. Today we call it a test cross because they do this still all the time, because it's a fast, easy way to see if an individual that shows the dominant phenotype has homozygous dominant genotype or heterozygous. So this purple one could be what for genotype? What's the possible genotype of this? Big A. We know that it's one of those two, but we don't know for sure which one. What's white? Yep. So it looks like this. If we're going to take white and cross it with purple, if it's big A, the gamete will always have a big A. For the white one, the gamete will always have a little a. There's only one box because the outcome is always going to be this. Make sense? If it's that way every single time, then all of the seeds planted from that would all be purple flowers. He would know, and we know now, that that purple plant is homozygous dominant. On the other hand, if the purple plant is heterozygous, the gamete will either have a big A or it will have a little a. The white flowered plant always gives a little a in the gamete, so then it looks like this. Now we have a 50-50 split. 1 out of every 2 are purple. 1 out of every 2 are white. So a test cross then is an easy way to know if your dominant phenotype individual is heterozygous or homozygous dominant. And he's the one that devised that so that he could take purple plants, and it was much faster now for him, cross it with a white, plant the seeds, so in a few weeks he would know whether that purple flower plant was big A, or big A, little a. In the past, remember, he had to do several generations. It took him months to figure out if a purple plant was homozygous dominant or heterozygous. So this was a lot faster for him. Even today we do that. We work a lot with fruit flies today for genetics, but they'll take a dominant phenotype fruit fly and cross it with a recessive phenotype. Okay, so by Wednesday, I want you to finish reading chapter 10 if you haven't already. The PowerPoints are online. Don't worry so much about the handouts yet, except for the one that says review for test 4. Okay, remember, this is really old. So this is not test 4 material. Not all of it. It's a good review. I want you to do the ones that have to do with mitosis and meiosis especially, just to review it. Okay, see you Wednesday. Oh, if you want to see your Scantron portion of the test, I did run those.
You want to eat?